Okay, so I'm gonna give you a little bit of a review on today's Graham Stanley lab, and I think people from last week must have told you that there will be a 20-question quiz at the end, and we'll have that in the workshop. And I'm trying my best to get it done by 4. I'm just waiting for Dr. Grimm. Well, let me give you a little bit of help on that. Firstly, remember that there are 4 videos on each and every those laptops or those computers over there for the drugs. So there are 4 questions from those straight questions from those videos. So they are important, right? And the rest of the stuff, which you had more time than the previous batch, you have 2 weeks to go through that because they were posted like 2 weeks ago. So other questions are coming from that. And I will give you some of the important aspects, and I expect you to know them because that will form the basis for that. Remember that today was lecture 7, and for every bacteria that I talk about, we talk about the cell wall, we talked about the antigen, we talked about something there, and almost all the bacteria will have you diagnose them, or at least initial diagnose them with Gram staining. So Gram staining pretty much is a gold standard, not because of the technique, but because of the understanding that you may have in terms of knowing what the outermost shell of bacteria is, what makes them survive, and what makes them vulnerable as well as to what drug are we going to use. All right, so that will form your basis of understanding the action of antibiotics. Okay? Now, I don't want to spend much time on history, but you can see that it took like maybe starting from 1928 when one of the lab person made a mistake in Professor Fleming's lab in England, that he failed to wash off or clean up the petri dishes, and when he comes back, he finds that there were fungus growing over there. And the very good thing he did was, as most of the researchers would do, is that instead of cursing the students or saying bad about the student, he thought of a good thing. He thought he observed something, that wherever those fungi were growing, there were absence of growth of bacteria. So he saw that kind of halos around those bacterial plates. So it just struck him that maybe fungi are releasing something which have an antibiotic action. So that is the basis of so-called antibiotics, which has changed the whole world. Not only that we are using antibiotics when you get sick, you are using antibiotics to the dairy animals, to your meat products, and you cannot imagine how much tons of antibiotics are consumed in this country. Tons of them. It's a big industry. So it took him at least 10 years, more than 10 years, to basically do all the lab work and experiments to really get it in a form that basically would be used as antibiotics. And after that, you can see 1940s, these are the Nobel Prize given to people, 1950, 1988, till 2009, when these 3 people are credited for something that is going on for us to use as antibacterial. Okay? Now, the question is that why do we need to do Gram staining? If you were to work in any place, even physicians, pharmacists, dentists, any healthcare professional, you see a person who is infected and you see a wound, you see some specimen, you immediately want to find out what kind of bacteria that is, because you want to start an empirical therapy. You want to start antibiotics, and you don't want to start broad spectrum antibiotics. You want to make at least this demarcation, gram positive versus gram negative, and look them under the microscope. So you want to start, we call it empirical therapy. Now, most of the drugs that you will see that are used today as an antibiotic action is that we want to stop bacterial division, because they are dividing maybe 2 every minute or second. We want to stop that. They are making their own cell wall. We want to stop the synthesis of cell wall. But we need to know how is it that they're making those cell walls. Where is this information coming from? No. So most of the lines of the drugs that you can see from here, which are cell wall active antibiotics, we call them beta-lactam antibiotics. And these are the most important, even penicillin as a drug of choice for many different things, right? Now, also remember that if the bacteria are already there, so you want to lyse the bacteria, right? You want to lyse the bacteria like we call it a lytic type of antibiotics, where you're not stopping the synthesis. We can only stop the synthesis in newly formed bacteria. So your drugs will go into the bacteria, and when they divide into two, they will not be able to divide it because you messed up the whole system, right? So there are different mechanisms that we are working with, and all of them basically are based upon the cell wall synthesis, and that's what the basis of today's lab as well. So the terms that we normally use, as I said, antibacterial spectrum, a broad spectrum which will have effect against gram-positive and gram-negative. We want to go for the narrow spectrum because we do have choices. Some of the drugs are very effective. They will have a bacteriolytic action. Some of them will be less effective. They may have bacteriostatic action. So there are many different, sometimes we do want to have one of them or both, right, depending upon where of the drugs are acting. And then we also want to find out how much is it that we need, because we don't want to overkill. You don't want to use too much of antibiotics. You remember where is it coming from? Because of disruption of normal flora. You don't want to do that. So you want to go for the minimum possible drug concentration. We call minimum inhibitory concentration. That's why you check on the plate, and you have to look at the dilutions and so on and so forth. Okay? Now, many times what we do is that when we define minimum bacterial concentration, MBC, we basically want to lower the concentration of the drug, but at the same time achieve the maximum benefit. We want to go for 100%. Don't think that, okay, we're going to lower this concentration and go for killing half of them. We don't want them because of the doubling time. Because if they are like 1 million, within a minute there will be 2 million. So there is a multiplicative progression of this bacteria. They're very fast. Okay? And sometimes what may happen is that we want to use more than one antibiotic. So for example, we want to stop the synthesis. We want to hold on to the nutrition requirements for the bacteria. We want to even stop the nuclear transcription that is happening. So we want to go for more than one choices. So we call it syllogism. So we use more than one antibiotic to go for the best possible solution, right? And sometimes they may have an opposite action. So they want to go, we call it antibiotic antagonism. So one is going in one direction, I'll explain that in a while, the other is going in opposite direction. We basically don't want that. But many times we have to do it. At those times we want to see what is the net result. What is the net result? If we end up having more beneficial effect, even going for antagonism, we can still go for that. Because we are targeting two different aspects of killing that particular bacteria. Okay? Now, you should expect, I think, maybe 5-6 questions on beta-lactamases. So you may want to spend some time on that. So this basically is, because remember we use antibiotics, but bacteria are clever as well. So they come up with their own packages and plan. So what they would do is that many of the antibiotics have beta-lactam ring. So bacteria will have an enzyme, beta-lactamase enzyme, that will chew that up, and thus will make those antibiotics ineffective. So we have a list of antibiotics, beta-lactam, and then the bacteria produces beta-lactamases, so the antibiotics are ineffective. So we have to go for another choices. So these kind of enzymes which are there, especially for penicillin or cephalosporin, which are the first line of the drugs, are also called penicillinases. Aces mean because they're going to mess up with the action of penicillin. And then there are a couple of other metallo-beta-lactamases. There is a chain of those enzymes. We need to know where they are present, how they work, and how can we fight them. Okay? And if any of you remembers, when we talked of outside-in signaling pathway in immune response, remember I said that something happening at the cell wall, and there is a series of events that will happen within the cytoplasm before nuclear factors will kick in and initiate the transcription of production of one particular protein. Right? And the whole sequence involves different set of enzymes. One of the enzymes, if you remember, we talked about serine proteases. So that's another word we call serine proteases. And you will see over here that most of the beta-lactamases they come from a large family which are serine proteases. And these basically are transpeptidases and transglycolases and carboxypeptidases. So the thing is, if you have a structure of that antibiotic, it has a carboxy ring, it has some other carbon link, each one of them will go and chew that up. And we need to know so we can stop that happening. Okay? And I think next slide you'll find quite useful. Is it a gram-positive or gram-negative?
Gram-positive.
Remember, I told you that gram-negative is like you have this double layer as compared to gram-positive. So you have a cytoplasmic membrane, right? And you have an outer membrane. So 2 loaves of bread. I gave you an example of the sandwich, if you remember. And then you have 1 cheese layer in between. So that's a peptidoglycan. Because in the gram-positive, you just have this half-cut loaf or bagel and just want to put big chunk of cream on that. So that's like a gram-positive as compared to a sandwich where there's a layer of one fatty layer of thin, transpeptidase and another on top of that. So outer layer and the cell membrane. And then the most important thing for gram-negative is that there is a periplasmic space. Periplasmic space. And this periplasmic space has those enzymes that we've been talking about, like beta-lactamases, right? Penicillin binding proteins, PBPs. So that's where the problem is. So if you give an antibiotic, it's going to be held in between 2 layers and would not even pass and do nothing to the bacteria. And then again, you have other important thing for the outer membrane, that there are pores. So there are pores over there. There are no pores, no outer membrane gram-positive. So any drug that you want to give has to pass through those pores. Okay? So the whole idea is that beta-lactamases that we talked about, if they are present, they reside in this space over here. And then penicillin binding proteins specifically, they are basically going all the way through and through into the cell membrane. So they're pretty much like stuck over there. Transmembrane proteins, that's what we call transmembrane proteins. They go through and through. So they have a inner edge and then an outer edge, and they are kind of stuck over there. And this is a typical gram-negative bacteria. We already studied that in the class, the structure of peptidoglycan, especially if this is Staph aureus one, you can see all these different type of chains. They are cross-linked peptidase bonds where you have different amino acid, and then different amino acids are binding with other chains. So that's another thing. There's one question on that as well. So there is an alternating band of these amino acids. And this is one of the actions that you will see in one of the videos presented over there. Very nice video. They will give you which particular antibiotic will go and break it apart, so that you basically disrupt the cell wall of gram-positive. So it's going to act on these one, or some of them are going to act over here. That's why we have different groups of antibiotics. Okay? Okay. Now, let's move on. This is just an explanation of what I already told you. The next question is that what are the mechanisms where beta-lactam antibiotics become resistant? So what are the resistant mechanisms, right? And there are many different mechanisms, but so far what we found out that bacteria have an ability to resist beta-lactam antibiotics by these three journal mechanisms. So these are three journal mechanisms. So bacteria, those bacteria that resist beta-lactam antibiotics, number one, you can see that they basically will prevent the interaction between antibiotic and the target penicillin binding protein. Okay, so they would not let that happen, because if the penicillin is targeted for penicillin binding protein, they would not allow them to reach there. That's number one. Number two is that what they will do is they will modify the binding. So that binding has to take place in a specific way to initiate cell killing. So they can modify that. If that's modified, the action would not be correct. And then third thing is hydrolysis of antibiotic by beta-lactamases. That was simple. So the moment you give that antibiotic, those specific lactamase enzymes, they hydrolyze. So these are the three journal mechanisms that you normally see. And the most important thing in this case is that the first one basically is only seen in Pseudomonas. This one is only seen in Pseudomonas. So Pseudomonas, if you know, is a very nasty bug in the hospitals to treat. And we have Pseudomonas in burn infection, Pseudomonas in lung infection. So we have a problem over there because they have an outer membrane that overlies the peptidoglycan layer. So that's the whole idea over here. Okay, we talked about porins. You can read it on yourself. What may happen in some of the other cases that you normally see, other than what journal mechanisms that I just described to you, specific mechanisms are, these are some specific mechanisms that we normally see in a specific bacterium. The other three that I told you, other than Pseudomonas, were general. But these are defined to a specific bacteria. Number one, basically, you have too much of penicillin binding protein, rare occurrence, doesn't happen. But second is important, where a new penicillin binding protein basically is there like staph. So what happens in this case is that bacteria, you make an antibiotic, a penicillin that should go and talk to the penicillin binding protein for an action. So what bacteria does it basically acquires a new penicillin binding protein, and whatever you made would never come and bind it to it. So that's basically a basis of MRSA, the problem in MRSA, which is methicillin-resistant Staph aureus. And you will see that typically where bacteria acquires a new PBB and you are not sure where antibiotics going to bind with that. And the third thing is that some of the bacteria will modify penicillin binding protein through recombination. So recombination is, again, just genetic techniques that we discussed earlier. Because remember, the bacteria are smarter as well. You are giving them antibiotic, and they are coming up with new ways and novel ways to resist that. And we are basically learning from them because they are smart enough to figure it out. So in this case, especially if they modify the existing penicillin binding protein, that happen in step pneumoniae, right? Or a point mutation that may happen Enterococcus faecium. So two important things. One is the recombination that we discussed as one of the three mechanisms. That happens in S. pneumoniae. The other one is a point mutation that happens in Enterococcus faecium. If you remember, one of the last slides was we have vancomycin-resistant enterococci, we call VRE. So some of the bacteria are MRSA, methicillin-resistant Staph aureus. VRE, vancomycin-resistant enterococci. So these are nasty bugs because they come and modify that up. So this is a specific information to that bacteria. Okay? And when we want to find out what kind of different beta-lactamases are, so you will see that, as I said earlier, they all are from the family of serine proteases, but there are tons of them. So we know at least more than 200. That's why the challenge is that some of them are specific for penicillin, some of are specific for those other cephalosporin, and so on and so forth. So every bacteria that we are discovering, they are coming and they are capable of inactivating most of the beta-lactamase antibiotics. So you can see the challenge that we are having these days because bacteria are changing. And not only that, we need to develop those new antibiotics, we want to know how does that change occur. So we can help out and find out what is it. Because if your penicillin is not working, if your cephalosporin is not working, you have no choice. And the highest mortality rate in anywhere, in any part of the world is because of infection. So you can imagine it's a big challenge over here, okay? Classes of beta-lactamases, there's something that you need to cram and remember. Class A beta-lactamases, basically they are found in these specific gram-negative rods. So this is a very specific gram-positive rod, which is a beta-lactamase A, and you can see it's a Escherichia and Klebsiella. So these are the two, especially if you are asked what kind of beta-lactamase is there in these gram-negative rods, they are class A. So these are the class A. I mean, you're going to hear about it so many times in the next 3-4 years. By the end of 4, you're going to remember it. So don't give me that look. Okay. And then there are extended spectrum beta-lactamases, and then you can see some of them are a bunch of them, especially when they come to be transmitted via plasmids. So you already know what the plasmids are. These are extra chromosomal circular DNA. So these are a bunch of those pieces that go and move from one bacteria to another bacteria. So before the bacteria dies, is killed by, so it passes information that, okay, I'm going to die, but this is a bit of information for my future progeny. If you ever come and fight this antibiotic, this is the information you need to survive. So that kind of information is passed by plasmids, and then there are tons of them. The good thing that we know is that many of them are zinc dependent. So you can see they are zinc dependent metalloenzyme. So this is something that we important because many a time you must have why the zinc is coming, why we have deficiencies of zinc in certain infection, because that zinc is being utilized by bacteria for their own growth. Okay. Class C basically have some of them, especially for cephalosporins, and it's easy to remember because cephalosporin, anything that ends with ases. So this particular beta-lactamase is going to chew up cephalosporins. Okay, because it may not seem important to you right now, but just imagine a person has osteomyelitis, very severe infection of bones. You try penicillin, you fail. The only choice left is cephalosporin. If you fail, what is it then you do? Nothing. See, so that's important. Okay, and then class D basically are there for beta-lactamases, penicillinases for gram-negative rods. So what I would do is that just make a little graph, and this class A, class B, class C, and at least I think there were 5-6 bacteria I just mentioned. Very easy for you to remember, but definitely there are a couple of questions on that. Okay, and last slide, you can also see good videos over there. So this is a bacteria, right? So you can see that this is the option we have. So you have a typical structure of this bacteria. This is a cell wall, a peptidoglycan cell wall. So this is a bunch of bacteria. We call them beta-lactams. They have this beta-lactam ring. They all are effective against the cell wall synthesis. Okay. Then we have other set of bacteria. For example, let's put this one, anti-metabolites, because if the bacteria have to divide, they need metabolites. They need nutrition. So what we do is that we stop that supply of nutrition to this bacteria. And this is we call anti-metabolites. So we can use both. We can even throttle them by squeezing their neck, but we can also stop nutrition over there. These are anti-metabolites. And then again, 2 of them basically act to stop protein synthesis, because remember, by the end of the day, all those enzymes, serine proteases and others are proteins. And all those structure elements in the cell wall is lipid, carbohydrate, and protein. So if you stop the transcription of protein, so you will have drugs that will act either at 30S or 50S. This is something you have to remember it for good. So this is a list of bacteria that stop 30S ribosome, and this is a list of bacteria that stop 50S rhizomes. So there are two different types of antibiotics, and you can see we normally use that. And then finally, we have some bacteria that act on DNA replication and some bacteria which act on RNA synthesis, especially for this one. You can see rifampicin, which is an anti-TB drug, and you saw today when I was mentioning Clostridium today, for almost all of them, I told you metronidazole. It's also called Flagyl. Those of you who know the other name of the drugs, and quinolones, they stop the DNA replication. So you can see from this flow chart that we do have a plan to stop the replication of bacteria, because bacteria is dividing every minute. It goes for binary fission, and then everything which is there from the enzyme, from the structure elements of the peptidoglycan, from the structure element of the outer layer, we want to stop either supply for that, protein synthesis for that, and DNA, RNA messages for that. And you can see at least 1, 2, 3, 4, 5, 6. So these are the 6 very common, basic modes of activity that we have to stop that happening. Okay? And then other slides, I'm pretty sure you must have read them earlier. You had 2 weeks to do that. As the other group can always say, they just had 1 week to do it, but for you had ample time in the world to do that. But I would still encourage you to make sure that you understand, because this will be the only lab that you will have so much time at your disposal to understand the cell wall, and you will see the cell wall by Gram staining. Why is it that it retains gram-positive dye? Why does it retain safranin? Why does it retain, iodine? So things of that nature, and what is the structure of that? Where is the protein element? Where is the carb element? And where is the periplasmic space? And what are these Betalactamases? So get used to these things, because this is going to be the very important thing. And today's lab also will give you a little bit of insight into the genetics as well. We'll have a genetics lab coming up as well. But the same process, what is it that caused bacteria to become resistant? You're going to be bombarded with this question over and over again. We really want you to understand, why are we having antibiotic resistance? How can we prevent it? If you were to go in all these R&D companies which are making new antibiotics, this is the minimum qualification they would want of you, that you know what are the mechanisms of antibiotic resistance. Simple questions. Okay? So I'm going to let you go and prepare for your labs. Good luck. Thank you.